Hello friends, good afternoon. Welcome to SAM Biology class. Friends, today we shall discuss about photoperiodism. Photoperiodism is the developmental responses of plants to the relative length of day and night. It is the ability of an organism to detect day length which makes it possible for an event to occur at a particular time of the year, thereby giving a seasonal response. Numerous plant species flower during the long days of summer. For many years, plant physiologists believed that the correlation between long days and flowering was due to the accumulation of photosynthetic products that occurred in long days. This hypothesis was later found to be incorrect by the works of two American plant physiologists, Whitman Garner and Henry Allard, in 1920s. They noticed that a mutant variety of tobacco, Maryland mammoth, grew profusely to a height of about 5 meters but failed to flower in the prevailing conditions of summer. However, the plants flowered in greenhouse during winter under natural light conditions. On the basis of these experiments, Garner and Allard concluded that the day length was the determining factor in flowering. They were able to confirm this hypothesis in different plant species and conditions. The term photoperiod refers to the period of time each day during which an organism receives illumination. This is the day length. The term photoperiod and photoperiodism was introduced by Garner and Allard. On the basis of photoperiodic requirements, they classified plants into different photoperiodic groups. They are: number 1, short day plants in which flowering occurs when days are short. This group includes qualitative short day plants which flower only when days are short, and quantitative short day plants in which flowering is accelerated by short days. The second group is long day plants in which flowering is promoted when days are long. This group includes qualitative long day plants which flower only when days are long, and quantitative long day plants in which flowering is accelerated by long days. The third photoperiodic group is day neutral plants which flower under any photoperiodic conditions. They are insensitive to day length. In such plants, the flowering is typically under autonomous regulation. In addition, there are other photoperiodic groups such as long short day plants which flower when they are exposed to a sequence of long days followed by short days. And the last group is short long day plants in which flowering is promoted when they are exposed to a sequence of short days followed by long days. This figure shows the photoperiodic responses in long day plants and short day plants. The essential distinction between long day and short day plants is that in case of long day plants, flowering is promoted only when the day length exceeds a certain duration termed as critical day length in every 24-hour cycle, whereas promotion of flowering in short day plants required a day length that is less than the critical day length. The absolute value of critical day length vary widely among species. Plants monitor day length by measuring the length of the night. Long day plants require a relatively short dark period for flowering. In fact, some of these plants flower best under continuous light. A long period of darkness has inhibitory effect on flowering in long day plants. In such plants, flowering is initiated when the long dark period is interrupted by brief flashes of light. Long day plants can also be induced to flower in short days provided that they are followed by still shorter night. For example, in case of a short day plant such as Xanthium strumarium, the critical day length is 15.5 hours. That means this plant requires a day length which should be less than 15.5 hours and a critical night of 8.5 hours of uninterrupted darkness for promotion of flowering. Another example is Hyoscyamus niger, which is a long day plant. In this plant, the critical day length is 11 hours. So, for promotion of flowering, this plant requires a day length which must exceed 11 hours and a relatively short dark period for promotion of flowering. This figure explains the photoperiodic responses in different groups. When the period of darkness is long, it inhibits flowering in long day plant, whereas it promotes flowering in short day plants. But when the light period is long and the dark period is short, it promotes flowering in long day plant, but the short day plant remain vegetative. In another experiments, when the long dark period is interrupted by brief flashes of white light or red light, then flowering is promoted in long day plant, but the short day plants remain vegetative. In another case, when the long dark period is interrupted by flashes of red light, or if the long dark period is interrupted by red light followed by far red light, then flowering is promoted in short day plant, but the long day plant remain vegetative. In short day plants, the flowering is inhibited when the dark period is interrupted by brief flashes of red light or white light. This phenomenon is termed as night break. Now, photoinductive cycle. It is the number of cycles of light and dark period required for initiation of flowering. Photoperiodic induction is a progressive process which yields a quantitative response. The number of photoinductive cycles provided is in excess of the minimum number required to induce flowering. For example, in case of Xanthium strumarium, the minimum number of photoinductive cycles for flowering is 1. Whereas for another plant such as Salvia occidentalis, it is 17. Both are short day plants. But in case of a long day plant such as Plantago lanceolata, it is 25. When a plant has been subjected to a minimum number of photoinductive cycles, it will flower even if it is placed under non-inductive condition. Next, perception of photoperiodic stimulus. In plants, leaves are the site of reception of photoperiodic stimulus. The photoinductive capacity varies with the age of the leaves. Generally, young leaves are less effective than fully expanded leaves. That means during photoperiodic induction, something is produced in the leaf and is then translocated via phloem to the floral bud, where it induces the formation of floral primordia. When a single leaf of Xanthium strumarium a short day plant is photoinduced, then the plant will flower. And when such a plant is photoinduced and is grafted to a series of non-induced xanthium plants, all plants will flower. So friends, this is all about photoperiodism. For more updates, please subscribe my YouTube channel, Sam Biology Class. Thanks for watching.